Hello everyone. Now welcome to another session of general embryology in which I will be discussing about the spermatogenesis, the structure of the spermatid, the spermatozoa, the blood testis barrier or also known as the Sertoli barrier. I will talk about the hormonal control of spermatogenesis and the sperm capacitation. So let's start. Now spermatogenesis. You already know what is the definition of spermatogenesis. Now let us first write the definition of spermatogenesis. Now spermatogenesis, it is the formation of spermatozoa or these are called as mature sperms. So it is the formation of spermatozoa or mature sperms from spermatogonium, right? From spermatogonium or spermatogonia, right? So this process, it is called as or it is known as spermatogenesis, right? So formation of spermatozoa or mature sperms from spermatogonium, it is known as spermatogenesis. Now this spermatogenesis, remember the spermatogenesis, it occurs in testis. Spermatogenesis, it occurs in testis, right? Now testis has two anatomical compartments. Now testis has two anatomical compartments. And these two anatomical compartments are, you already know, we have the interstitial compartment, and the seminiferous tubules, right? Now spermatogenesis will take place in seminiferous tubules. Spermatogenesis occurs here, right? Now this is the diagram which is depicting the testis, and you can see these seminiferous tubules. The testis is divided into various lobes, and these are called as testicular lobes, right? And these testicular lobes are containing these tubules, highly coiled tubules, and this is called as the seminiferous tubules, right? We have various lobes in the testis, and these lobes are containing these seminiferous tubules, and in between we have the interstitial compartment or the interstitial space, okay, that you can see in between these compartments, right? Now seminiferous tubules basically they contain two types of cells, right? They contain the Sertoli cells. From S, you have to remember Sertoli cells. These Sertoli cells are also known as sustentacular cells. These are also known as sustentacular cells, and the seminiferous tubule will also contains the germ cells, the spermatozoa, spermatogonium, right? Primary spermatocyte, secondary spermatocyte, all this will be in the seminiferous tubule. Why? Because spermatogenesis is taking place. So from spermatogonia till spermatozoa, the various stages of development, okay, so you will find all the germ cells. So all the germ cells in various stages of development, in various stages of development. So we will have germ cells in the various stages of development. Now, apart from that, you should remember that in the interstitial compartment, you will see one cell that is called as the Leydig cells. You will see Leydig cells. Okay. You will see fibroblast, and you will see the neurovascular tissue. Okay, neurovascular bundle, it means the nerves and the vessels. Right? So this is about the anatomical compartments of the testis, and our point of area, or our area, it is like the seminiferous tubules, right? Now, firstly, before starting with the spermatogenesis, let us just first understand what is the pathway. Okay, what is the pathway of, like, you already know over here that the testis. Now let us just draw a flowchart. A certain basic points that I want to tell you before starting with the spermatogenesis. So you already know we have the testis, right? Now this testis contains various lobes. Now it contains various lobes. Now these lobes, right? These lobes are called as seminiferous tubules. These lobes are containing these seminiferous tubules, right? You can see these highly coiled seminiferous tubules are there, right? These coiled seminiferous tubules. Now, these seminiferous tubules will form these straight tubules, which I am highlighting right now. These are the straight tubules which will be formed, right? These are the straight tubules. Straight tubules. So these seminiferous tubules, they form straight tubules, right? They form straight tubules, and these straight tubules, you can see over here that I am highlighting, they are joining together. The yellow color which I am highlighting, they are joining together to form the rete testis, right? They are joining together to form this yellow color which I have highlighted. It is the rete testis. Rete testis. So these straight tubules, basically they join and forms rete testis, right? They join and form these rete testis. Now, from these rete testis arises efferent ductules. So if you can clearly see over here, the one which I will be marking with red color, yes, this one, yes, these are the efferent ductules. You can see these long cord like projections. These are like efferent ductules. Now these efferent ductules will open into epididymis. This will open into epididymis. Now you can see the epididymis over here, this long part, right? This long part, it is the epididymis. So these all tubules are like opening into this long structure that is called as the epididymis. Epididymis. Now this epididymis contains the head part or the cranial part, okay, the head or the cranial part, and we have the tail part of the epididymis or the caudal part of the epididymis. So this epididymis has, remember, we have the head part, head or the cranial part, and we also have the tail or the caudal part of the epididymis. Caudal part of the epididymis. Now this epididymis will opens into vas deferens. It opens into vas deferens. So you can see over here, this is the vas deferens, right? The vas deferens, right? So they open into the vas deferens where you will see right now over here. So you can see that in this particular picture that this is the, let me just use black color, this is the testis, right? This is the epididymis. From the epididymis, this is the vas deferens, right? This is the vas deferens. Now this vas deferens, we have the seminal vesicles, okay? We have the seminal vesicles, which will pour its secretion, okay, via seminal tubule, right? Via seminal tubule, the seminal vesicles will pour its secretions, right? Then we have the prostate glands which will pour its secretion, and finally it will be, all the things will be going into the ejaculatory duct. You can see over here, this is the ejaculatory duct which will open into the penis. Okay, at the time of the intercourse, there are other glands such as bulbourethral glands will pour its secretion. It is only at the time of intercourse. Otherwise, it is the seminal vesicles and the prostate glands which will pour its secretion into this vas deferens, and this vas deferens will receive the secretions from the seminal vesicles, prostate glands, and ultimately it will land up into the ejaculatory duct. So I hope this is clear, the pathway is clear. So now let's talk about the spermatogenesis. Now spermatogenesis basically takes place at the puberty. Now it will take place at puberty, right? It is the formation of mature spermatozoa. From the spermatogonium. तो चलो spermatogonium बनाते हैं पहले. You already know this is, for example, let us suppose this is the spermatogonium. Spermatogonium, which contains 46 chromosomes. So we write it as 46XY, or we can also write it as 44 plus XY, right? It is 46XY or 44 plus XY, right? So yes, we can just write it in the cell also, like it is 44 plus XY, right? Now this spermatogonium will differentiate into, right? It will differentiate into two types. We have dark spermatogonia and pale spermatogonia, right? So we have two types over here. We have dark spermatogonia and pale spermatogonia. Now, dark spermatogonia, it is type A spermatogonia. And pale spermatogonia, it is spermatogonium, it is also known as spermatogonia, right? Spermatogonium बोलते हैं, singular plural spermatogonia, right? So pale spermatogonia, it is type B spermatogonia. Now, this was a question that has been asked. Which spermatogonia undergoes mitosis? It is type B spermatogonia, right? Then what is the function of type A? Now, type A or the dark spermatogonia will keep undergoing mitosis until the stores are replenished, right? Until the stores are replenished, मतलब जितना चाहिए उतना अमाउंट में बन जाए, replenished, right? So they will undergo mitosis and they will form spermatogonium only until the requirement is done. So they will continue to divide and form spermatogonium only. Now, type B spermatogonium, it will undergo mitosis. Okay. Sorry, the question was like which one will undergo mitosis to form the primary spermatocyte. So, yeah, type B spermatogonia will undergo mitosis to form the primary spermatocyte. Okay, it will form the primary spermatocyte. Now, primary spermatocyte, it is like big, so it is containing all the growth factors and all the nutrition, so it is like big and large in size. Now, if there is mitosis in the spermatogonium or spermatogonia, what is formed? Primary spermatocyte. Again, after mitosis, the chromosome number will remain the same, that is 44 plus XY, the chromosome number will remain the same, right? That is 44 plus XY, right? Now, this primary spermatocyte, so I will draw it on the next slide, but you will continue with the same flowchart, right? Now, this primary spermatocyte, so I will just mark this over here, yes. This primary spermatocyte will undergo first meiotic division, right? It will undergo, I'm just drawing like this, you have to continue. It will undergo the first meiotic division. Now, first meiotic division, I have already told you, it is a reductional division. It is a reductional division, right? It is a reductional division where the chromosome number will be reduced to half. So, the resulting secondary spermatocytes will have 23 chromosomes each, right? So, this is the secondary spermatocyte. Secondary spermatocyte are formed from the primary spermatocyte. How? By the first meiotic division, and the resulting secondary spermatocyte will have the haploid number of chromosomes, that is 22 plus X and 22 plus Y, right? 22 plus X and 22 plus Y. So, these are the secondary spermatocyte. Now, these secondary spermatocyte will further undergo, secondary or second meiotic division. Okay, they will further undergo the second meiotic division, okay, and they will form spermatozoa, the round globular structures. These are called as spermatozoa, So these are called as last two lines repeat. Now the secondary spermatocyte will undergo secondary meiotic division or second meiotic division to form round globular structures called as spermatid. Right? These are called as spermatid. Now these spermatid will again, second meiotic division, I have already told you it is like mitosis, so it is like equational division. The number of chromosomes will remain the same, that is, yes, over here it will be 22 plus x, 22 plus x, and we have 22 plus y, 22 plus y. 50% of the cells will have 22 plus x and 50% will have 22 plus y. Now these spermatids will undergo differentiation and maturation. Okay, this will undergo differentiation and maturation. Now there is no mitosis or meiosis. There is only differentiation and maturation at this stage, and they will form sperms or spermatozoa. They will form sperm or spermatozoa, the tail. So these are called as the mature sperms or spermatozoa. Okay, these are called as mature sperms or spermatozoa, right? So yeah, so formation of spermatozoa from spermatogonium or spermatogonia, it is called as spermatogenesis. So this was about the spermatogenesis. Now spermatogenesis basically takes place in the, you already know the answer, it is the seminiferous tubules. It is the seminiferous tubules. Now the time taken for spermatogenesis, again a very important question, it is 72 to 74 days. It is 72 to 74 days. And remember, what is spermiogenesis? Spermiogenesis it is the transformation, okay, it is the transformation of spermatid, okay, transformation of spermatid into sperms. So the last step that we have discussed, where there is no mitosis or meiosis taking place. So I hope everyone can see this. So the spermatid transforming into mature sperm or spermatozoa, this process it is called as spermiogenesis. This process it is called as the spermiogenesis. So remember, yeah, over here one more point that we have to remember, that there is no mitosis or meiosis. There is no mitosis or meiosis, right? And the time taken, very very important, the time taken over here, it is 14 days. The time taken over here, it is 14 days. Time taken for total spermatogenesis, it is 72 to 74 days, and the last step, that is conversion of the spermatids into the sperms, it is spermiogenesis, which is almost taking around 14 days to complete. Let us draw the structure of the spermatid. Now, spermatid, it is a round globular structure. It is a round globular structure, right? Now, this spermatid, it is containing in center, it is called as the nucleus. It is called as the nucleus, and this nucleus will contribute, okay, it will contribute to form the head of the sperm. It will contribute, or it will transform to the head of the sperm. It will contribute or transform to the head of the sperm, right? Now, we have various mitochondria over here, right? In this spermatid, we have mitochondria, numerous mitochondria are present. Now, this mitochondria will transform into the middle piece of the sperm. M से M याद रखना, it will transform into the middle piece of the sperm. It will transform into the middle piece of the sperm. Now, there is something which is called as the Golgi complex. Now, there is something which is called as the Golgi complex. Now, this Golgi complex will form the acrosomal cap, and we have two centrioles that are present over here. The two centrioles. Now, we have these two centrioles. One centriole will transform, or it will transfer, or it will transform into the neck region, while the other centriole will transform, okay, with the tail region. Other centriole to tail region, right? Now, this tail region contains various axial filaments, right? So, this tail region will contain various axial filaments. So, this was about the structure of the spermatid. Now, coming on to the next, that is the structure of the sperm. Now, structure of the sperm or the spermatozoa. So, a very simple line diagram that I will draw for you people. You already know that this is the head of the sperm, right? This is the head of the sperm. The head continues with the neck of the sperm. Then we have the body of the sperm, which contains the various mitochondria, right? And at last we have this tail of the sperm, right? So remember that this is the head, this is the head, this is the neck of the sperm. The head also contains the acrosomal cap, right? The acrosomal enzymes. Now, in the body of the sperm, we have these mitochondria, right? So, this is the middle piece, I can say, the middle piece of the sperm, and these middle piece is containing these mitochondria, which provides energy for the motility, right? Which provides energy for the motility. Then we have this tail part, and in the tail part you can see that we have this axial filament. And remember, this axial filament, it contains a protein that is called as the dynein protein. And remember, this dynein protein, it is very important. It plays an important role in the motility of sperm. So remember, this is an important point, that this dynein protein plays an important role in the motility of the sperm. It plays an important role in the motility of the sperm. Now, size of the sperm. Now, the size of the sperm, it is 50 to 60 microns. It is 50 to 60 microns. The fertilizable span of the sperm, it is around 48 to 72 hours. It is 48 to 72 hours. Sperms are formed in seminiferous tubules. Seminiferous tubules, and you already know they are non-motile and immature. They are non-motile and immature. They are emptied into a network of ducts called as the rete testis, right? I have already discussed with you people right now a couple of minutes back. So yes, it is the rete testis, and from the rete testis, the sperms will reach the epididymis. From rete testis, the sperms reach the epididymis, right? Now, the epididymis contains the head part, also called as the cranial part, or also called as the proximal part of the epididymis, and it also contains the tail part, also called as the caudal part of the epididymis, or also called as the distal part of the epididymis. Now, the proximal part is the part or the head part where the sperm will attain the maturity. Head में देखो mature बनता है ना head. So the sperms, it attains maturity, and the tail or the caudal part is the one where the sperm will attain motility. The sperm will attain motility, right? So yes, now there is a protein that you have to remember that is called as the beta defensin protein. Now this protein, it helps in the attainment of the maturity, right? Or sorry, motility. Beta defensin protein, so it helps in the attainment of the maturity, motility. Attainment of motility. Two different questions. Maturity head वाले part में आएगी और tail वाले part में क्या आएगी? The motility, right? Now, epididymis acts as a reservoir of sperms until ejaculation. ठीक है? Now, the time taken for the epididymal maturation, it is about 2 to 14 days, right? It is nearly about 2 to 14 days. Epididymal maturation takes about 2 to 14 days. Now coming on to the next topic, that is the blood testis barrier. Now this blood testis barrier is actually a misnomer. It is actually a misnomer. It is called as the Sertoli barrier. It is called as the Sertoli barrier. Now, if we take the cross section of a seminiferous tubule, now I am taking the cross section of seminiferous tubule. Now what I will see in the cross section, just see over here, that let us suppose this is the cross section. Let us suppose this is the cross section, and in this cross section what you will find that there are Sertoli cells. Now the Sertoli cells are like the cuboidal cells with big appendages. Now these are the Sertoli cells with like finger like appendages are seen over here, right? So yes, these are the Sertoli cells. Now These are the Sertoli cells. Now these Sertoli cells are cuboidal cells, okay, cuboidal cells, and these cuboidal cells are having appendages, right? These are having appendages. So yes, these are the cuboidal cells having appendages. Now we have the lumen in between, right? And we will see, yes, we will see the various germ cells, the various cells which are like developing, the sperm cells, the mature sperm cells, spermatozoa. We will see the various sperms, immature cells around the periphery, right? So yes, so now you can very well see that there is a junction between the Sertoli cells and the Sertoli cells. Over here, if you clearly see that I am talking about this junction, this is the tight junction, and this tight junction, it is between the Sertoli cells. It is between the Sertoli cells. This tight junction, it is called as the blood testis barrier. It is called as the blood testis barrier. Now, it is a misnomer. There is no blood testis barrier. You can see that these junctions are present in between the Sertoli cells. So this is also known as the Sertoli barrier. Now, why this junction is important? Because it prevents the germ cells. Now, it is preventing the mature germ cells from the toxins that are present in the blood. The toxins in the blood, they could not reach the germ cells and damage them. So for the prevention of these toxins from the blood vessels, they are like forming these tight junctions, right? They are forming these tight junctions. So if you can see, if I want to draw some other image for you people, like for example, let us just see over here. You already know we have these, let us suppose these cuboidal cells, or these are the Sertoli cells. These are forming the tight junctions. Yes, these are the Sertoli cells, and inner to the Sertoli cells, you can see we have the various germ cells in the various stages of development. So we have germ cells, yes, we have germ cells. Let us like draw, these are the germ cells, and outside the Sertoli cells, we have the blood vessels, right? We have the blood vessels. Now, these blood vessels, whether it is arteries or whether it is veins, they don't allow the toxins present in the blood to reach the germ cells, so that they could not harm the germ cells. See how nature has given this is so important. Okay, so it is preventing the germ cells and to reach this, preventing the toxins present in the blood vessels to reach the germ cells. And outside this blood vessels, you will see another cells over here, and these are called as the Leydig cells. These are called as the Leydig cells. Leydig cells. So remember, the Sertoli cells, it is preventing the germ cells from toxins present in the blood vessel, in the blood. Okay, so it is acting as a barrier between the germ cell and the blood vessel, right? It is acting as a barrier between germ cell and blood vessel. And one more thing that you have to remember, the cells which lie outside the blood testis barrier it is the Leydig cells, right? It is the Leydig cells, right? It is lying outside the blood testis barrier. This was regarding the blood testis barrier. Now let us move on to the next topic, that is the hormonal control of spermatogenesis. Hormonal control of the spermatogenesis. Now you already know we have a structure that is called as the hypothalamus, right? We have a structure that is called as the hypothalamus. Now this hypothalamus releases GnRH. Now GnRH stands for gonadotropin releasing hormone. It is gonadotropin releasing hormone, right? Now this hypothalamus, which is releasing the gonadotropin releasing hormone, it will act on anterior pituitary. It will act on anterior pituitary. Now anterior pituitary will release two hormones. Now it will release two hormones. One is FSH and the other one it is LH. FSH stands for follicle stimulating hormone. Follicle stimulating hormone. And LH stands for luteinizing hormone. Right? The follicle stimulating hormone will act on the Sertoli cells. देखो FSH में H आता है. It will act on Sertoli cells. Okay, it will act on the Sertoli cells, and these Sertoli cells will release inhibin B. A hormone that is called as inhibin B is released. Now this inhibin B will have a negative effect on FSH, right? It will have a negative effect or negative feedback. It will have a negative feedback on FSH. So FSH is acting on the Sertoli cells, and these Sertoli cells will be releasing inhibin B, which will have a negative feedback on FSH, right? On the other hand, the luteinizing hormone will act on the Leydig cells. So you already know LH. L stands for, over here, like you can remember, it is the Leydig cells. The Leydig cells, and these Leydig cells will release testosterone, and this testosterone will bring out secondary sexual characters, and it will also support spermatogenesis. It will also support spermatogenesis. Even the FSH and the LH both will support spermatogenesis. Now if I ask you the major support for spermatogenesis, it is testosterone. The major support for spermatogenesis. Now let us write these questions also. So yes, we have the major support for spermatogenesis. It is from the testosterone, right? LH and FSH, they also they also support spermatogenesis. They also support spermatogenesis, right? They also support spermatogenesis. So if I ask you a question, all the hormones support spermatogenesis except, so except koi bhi ho. So you all know the three, the testosterone, the follicle stimulating hormone and the luteinizing hormone. Apart from this, if any other option is given, you can just mark it as the answer. So yes, if LH, FSH and the testosterone, they are not present, okay, if normally these three hormones, testosterone, LH and FSH, if it is not present in normal amount, right, then it can lead to defective spermatogenesis. It can lead to defective spermatogenesis, right? And this can result in azoospermia. Azoospermia, I will be discussing this azoospermia in the upcoming part, right? So it can lead to the defective spermatogenesis, and that could be a reason for the azoospermia. Now coming on to the last topic, that is the sperm capacitation. Now, what is the definition? Sperm capacitation basically stands for the ability of the sperms to fertilize the ova. Ability of sperms to fertilize the ova. Okay, you already know the sperms are stored in the epididymis, are mature, but they cannot fertilize ova. The final step that will make these sperms capable of fertilizing the ova, it is called as this capacitation. The time taken for capacitation, it is around 7 hours. Okay, approximately the timeline, it is 6 to 8 hours. Okay, capacitation occurs, or I can say the sperms will attain, okay. Now first, you already know the sperms attain motility in which part of the epididymis? It is the distal part of epididymis, or the caudal or the tail part of the epididymis, right? Epididymis, right? Sperms become hypermotile in the cervix. The sperm becomes hypermotile in the cervix. So cervix is the place where capacitation will take place. So remember that cervix, it is the place where capacitation will take place, right? Capacitation will occur, right? In the female reproductive tract, it is the cervix. Apart from that, remember the vagina, okay, they act as a reservoir, okay, for sperm for about 2 hours, right? They act as a reservoir for sperm for about 2 hours, okay, and cervix can also act as a reservoir for 3 days, right? If sperms are like they remain in the vagina for more than 2 hours, they become immotile, right? They become immobile, they become immotile. So that is about the sperm capacitation. Thank you.